Hi friends, welcome back to my channel Only Bio. I'm continuing with the series of the challenging MCQs in biology, Group A from number 16 to 20. All of these questions will be very useful for different competitive exams like NEET, AIIMS, PMT, AFMC and so on, and all these are with the detailed explanation. So question number 16 relates with the chapter that is human health and diseases. The question is, there are 5 classes of immunoglobulins and only IgG can cross the placenta. This differentiation is on the basis of dash. And your options are A. Molecular weight, B. Light chains, C. Electrophoretic mobility, and D. Sedimentation coefficient. And the correct answer for this particular question is C, that is electrophoretic mobility. Friends, remember that immunoglobulins are nothing but commonly called as antibodies. These are chemically glycoprotein molecules. Please remember glycoprotein molecules that are produced by the plasma cells that are nothing but the WBCs as an immune response. They have the capacity to bind to a particular antigen and bring about their destruction. So in simple words, you can say that immunoglobulins form a part of humoral immune response. Now, there are 5 major antibody classes. Those are IgA, IgD, IgE, IgG and IgM. Okay, now this classification is based on differences in the amino acid sequences in the constant region of the antibody heavy chains. Perhaps you might recollect the structure of the antibody, the general structure, right? So IgG जो होते हैं उसमें the heavy chains are nothing but the gamma chains. Hence they are called as G, that is G for gamma. IgM, they have the heavy chains which are called as mu chains. IgA have alpha chains. IgE have epsilon chains and the IgD have delta chains. Now of this, it's only IgG which is predominantly present in the human serum, almost up to 75% of the total serum. And remember, IgG is the only immunoglobulin that can cross the placenta in humans and is largely responsible for protection of the newborn during the first few months of life. And because it's relatively abundant and has an excellent specificity towards the antigens, IgG is principal antibody used in immunological research as well. Now, IgG कहाँ होता है? So it is the major immunoglobulin that can be found in the blood, in the lymph, in the cerebrospinal fluid, in the peritoneal fluid, and it forms a very, very important factor in the humoral immune response, as I did tell you. And IgG की और एक खासियत है, that is it can be retained in the body for a long time. Okay, now IgM जो होता है, IgM is produced at the first sight of a threat. So think of IgM as the first wave of soldiers sent into the battle to assess the situation and to recon, and it forms almost 10% of the total serum, human serum, I mean immunoglobulin content. Okay, so it is the first antibody built during an immune response. Very, very important to be remembered. Now, IgA जो होता है, you can look on the screen because you can see something about the immunoglobulins here. IgA works as a surveillance at the surface barriers. For example, the lining of the gut and the respiratory system. These are the surface barriers. So, वहाँ पर we find the presence of IgA. प्लीज ये बहुत इंपॉर्टेंट है, आप लोग याद रखिएगा. IgD, IgG I have already told you. IgD, it exists in the small amounts in the blood and it is the least understood antibody. And IgE is typically used to determine if you have any allergy. So these are the important points to be kept in mind for the immunoglobulins. We now come to the next question, which is an additional question. Friends, as you all know, there are many questions which are the additional questions, which are related, no doubt, with the topic, but they are not really directly from the textbook. The question is, The bacterial culture of Alexander Fleming during his discovery of penicillin was dash. And your options are A. Staphylococcus aureus, B. Penicillium notatum, C. Streptococcus, and D. E. coli, that is Escherichia coli. Now friends, any guesses? I'm sure when you talk about the discovery of the penicillin, but obviously आपके दिमाग में जो आंसर strike होने वाला है, that is the option B. And you'll be surprised to know that they've asked you the bacterial culture, not the source of penicillin. So the answer is not B. You will be surprised here. The answer is A. Friends, remember Alexander Fleming was a Scottish physician who was the first to suggest that a penicillium mold secretes an antibacterial substance. No doubt he named it as penicillin, the first modern antibiotic, you can say, from 1928. And इसके बाद करीबन 12 साल Fleming grew them, distributed them and studied the original mold, which was determined to be a very, very rare variant of Penicillium notatum, and it is now known as Penicillium rubens. Okay, so now we go to the next question. Now the next question relates with the chapter evolution indirectly, and the question is, Hinny is the hybrid of dash, and your options are A. Stallion and female ass, B. Jack and mare, C. Jack and female zebra, and D. Lion and panther. And your answer is A. Stallion and female ass. You can look on the screen to see how an innocent Hinny looks like, right? It is a domestic hybrid, and please remember it's the offspring of a male horse. A male horse is called as the stallion, and a female donkey is called as the jenny. Okay, it is a reciprocal cross to the more common mule which you have heard of, which is a product of a male donkey called as the jack and a female horse called as the mare. Okay, we come to the next question. The next question relates with the chapter sexual reproduction in angiosperms. And the question is, in Arachis hypogea, A. Cleistogamous flowers grow below the soil. B. Chasmogamous flowers grow below the soil. C. The pedicel of cleistogamous flowers lengthens after fertilization and penetrates the soil. And D. The pedicel of chasmogamous flowers lengthen after fertilization and penetrate the soil. And the correct option for this question is C. Okay, now you can look on the screen how does exactly this Arachis hypogea look like. Friends, Arachis hypogea is nothing but the ground nut or the peanut, at times very crazily even called as the monkey nut. It belongs to the family Fabaceae, okay, which is also known as the Leguminosae or the pea family. Now, peanut pods jo hote hai, they develop underground, an unusual feature which is known as geocarpy. I repeat, geocarpy could be asked to you in a different form in the MCQs, right? And after fertilization, what happens is a short stalk at the base of the ovary, which is termed as the pedicel, elongates to form a thread-like structure known as the peg. And this peg grows down into the soil, and the tip, which contains the ovary, develops into a mature peanut pod. The pods are almost 3 to 7 centimeters long normally, and they contain 1 to 4 seeds. Hope you understood this. The next question relates with the chapter photosynthesis, and the question is PEPCO. PEPCO, I hope you can recollect, is nothing but phosphoenol pyruvate carboxylase, yeah, at times called as PEP carboxylase, yeah, PEPCase or PEPC, right, has an advantage as compared to Rubisco. I hope you remember Rubisco, ribulose biphosphate carboxylase. The advantage is that, and your options are A. Rubisco fixes carbon dioxide only in C4 plants, whereas PEPCO that is phosphenol pyruvate carboxylase, PEP carboxylase, does it both in C3 and C4 plants. Your option B, Rubisco is subject to photorespiration but PEPCO is not. C, PEPCO is present in both mesophyll as well as in bundle sheath cells, but Rubisco is not. And D, PEPCO can carry out both anabolism and catabolism, but Rubisco cannot. Friends, please remember PEPCO is nothing but फॉस्फोइनॉल पाइरुवेट कार्बोक्सिलेज या हम जिसे शॉर्ट फॉर्म में पीईपी कार्बोक्सिलेज कहते हैं. And the correct answer for this question is B. Rubisco is subject to photorespiration but PEP carboxylase is not. Friends, remember PEP carboxylase is an enzyme in the family of carboxylases, right? And it helps in the addition of bicarbonate to phosphoenol pyruvate to form the 4-carbon compound that is oxaloacetate and some inorganic phosphate along with it. Now, this reaction is used for carbon fixation mainly in the CAM and the C4 plants. Now, CAM plants आपको शायद याद हो crassulacean acid metabolism जिसमें होता है. So, those plants which follow the CAM pathway are called as the CAM plants, and the C4 plants to regulate the flux through the citric acid cycle also in bacteria and plants. Okay, please remember, and C4 plants have carboxylating enzymes that have a higher affinity for carbon dioxide compared to Rubisco, and especially at the low carbon dioxide concentrations and high temperatures. The C4 pathway uses PEP carboxylase, which has a much higher affinity for carbon dioxide than Rubisco. In addition, PEP carboxylase does not have an oxygenase activity. So you can look on the screen very clearly to understand the difference here. Rubisco and how does Rubisco function actually, right? So this is what is it? In presence of low temperature and high carbon dioxide to oxygen ratio, it performs the Calvin cycle. And in case of high temperature and low carbon dioxide to oxygen ratio, it goes for photorespiration, which really is never going to be good for the plant because it utilizes or it destroys the fixed carbon, right? This is what conversation for the day. Thank you so much for being. And yes, please like, subscribe, and hit the bell icon and share this with maximum number of your friends so that we all can study together.